Evening viewer, welcome to our Glory Globe TV. Here's some amazing news. Efforts to develop a gene therapy for Diamond Blackfan anemia, that is called DBA, a rare, life-threatening disorder in which bone marrow cannot make mature, functioning red blood cells, have been hampered by the fact that at least 30 different genetic mutations can cause the disorder. A team led by researchers at Harvard Medical School has now cleared that obstacle by developing a universal gene therapy for DBA, one designed to correct the bone marrow defect no matter the patient's specific mutation. The experimental therapy is ready to test in clinical trials, the team reports November 11 in Cell Stem Cell. This is one of the first examples where we can develop a gene therapy that can target dozens of mutations with a single vector, said senior author Bijoy Shankaran, the HMS Jan Alien Paradise MD professor of pediatrics at Boston Children's. The work provides proof of concept that gene therapies can treat rare or complex blood diseases not by correcting individual mutations but by targeting a problem that all the mutations lead to, the team said. Children with DBA, which was first described in 1938 at Boston Children's, have few treatment options. A handful who have well-matched donors can be cured with bone marrow transplants, but most rely on steroids, which have side effects, or regular blood transfusion. Most of the gene mutations known to lead to DBA were discovered at Boston Children's over the past 15 years by Shankaran and Hannah Gazda. These mutations mostly affect ribosomes, structures within cells that play a key role in building the body's protein. But in 2012, as an intern in pediatrics, Shankaran found that a few patients instead had mutations in a gene called GATA1, a key regulatory factor that controls the earliest steps of red blood cells production. Shankaran subsequently found that the ribosomal mutations reduce the number of functional ribosomes in cells, which prevents GATA1 protein from being produced. When Shankaran added GATA1 protein back to blood stem cell taken from patients with DBA, the cells were better able to differentiate into red cells. These findings suggested that the raising GATA1 levels could treat DBA in patients with mutation in the GATA1 gene itself and in patients with the ribosome related mutation. Behind this exclusive report.